Hello everybody, welcome to Ms. Magvar video lecture, and today we will cover patterns of inheritance that beyond Mendelian's law. Learning outcome for this chapter. After studying this chapter, students should be able to explain the effect of lethal alleles on Mendelian ratio, distinguish among complete dominance, incomplete dominance, and codominance, distinguish epistasis from interactions of alleles of the same genes, describe the mode of inheritance of a mitochondrial trait, explain how inheritance of linked traits differ from inheritance of genes on different chromosomes, discuss the basis of linkage in meiosis, and explain how linkage is used to derive genetic maps. Now, this video lecture will be divided into two parts. The first part, we pretty much cover up to this mitochondrial inheritance. Oh, why it's 7, 8, 9, 10? I'm sorry, this need to be changed. So this need to be 4, 5, 6, and 7. Right, so in the first lecture, we will cover up to a mitochondrial traits, and the second part of video lecture covers the rest of the learning outcome. Okay, let's go ahead and begin. When gene expression appears to alter Mendelian ratios, so Mendel's traits showed two distinct forms. If you remember from the previous lectures, when Mendel studied patterns of inheritance, he used garden pea plants, and he chose plants that has distinct characteristic. For example, he would cross tall plant and a short plant. He would cross plant that produces green seeds with plants that produce yellow seeds. So that's what it means. He used two distinct characteristics. However, most genes do not exhibit simple inheritance. So very often, if you cross some plant that has green seeds and yellow seeds, all of a sudden you will have light green or dark yellow. Or if you cross tall and short plant, all of a sudden you have plants all in the medium height, right? Or all in between. So how this can be explained? Well, this chapter considers three general phenomena that seem to be exception to Mendel's law, but actually not. This is very important. We're going to look at the traits and the inheritance. It might seem that it does not follow the Mendel law, but it's not true. They do follow Mendel's law. And these three phenomena include gene expression, mitochondrial inheritance, and linkage. And again, in the first part, we will only cover gene expression, how gene expression seems to contradict Mendel's law, but it actually doesn't. In several circumstances, phenotypic ratio appear to contradict Mendel's law, but they do not. And the first example will be lethal alleles. Now, lethal genotype causes death before individuals can reproduce, and it removes an expected progeny class from a specific cross. For example, a double dose of dominant allele may be lethal. This happens in disease called genetic condition, I would say, called achondroplastic dwarfism. Achondroplastic dwarfism caused by a gain-in-function mutation in the gene FGFR3 that normally inhibits growth of the long bones of arms and legs. So what we're gonna look at in a minute, that to calculate the ratio of the traits that are expressed in offsprings. So for example, if you have tall plant and a short plant, you cross them, and now you need to count, well, how many tall plants you have when this tall and short reproduce, right? So you cross them, you have, let's say, 100 seeds. Then you're gonna plant this 100 seeds, and then you will count, well, how many of this, out of this 100 seeds grow tall? How many of these 100 seeds grow short? But what if some seeds are never sprout, so they just don't give any offsprings? Why? Well, maybe something wrong with this with those seeds, right? Maybe something wrong with genes that doesn't allow embryo to develop. So that might be lethal alleles that will remove that expected progeny. And then your calculation is not taking it into consideration. So let me see, let's look to the next slide to make it more comprehensible. So first, this is how achondroplastic dwarfism look like. You all probably know Peter Dinklage. This guy he is a very, very famous actor who played in Game of Thrones movies. He has this dwarfism. So you see how his head and his body are normal proportions, but arms and legs are shorter. Now, this is phenotype, but let's look at genotype of this genetic condition. Here we're gonna use little a as a normal height allele. So this is wild type, means most common in the population. And a big A is achondroplasia. That's a mutant gene. And this is what give that protein abilities to, or that genes that have this mutation. So gene that has a mutation that we abbreviate capital A, subscribed and translated into proteins that will inhibit growth of arms and legs. Now, those are our alleles. Remember, every person has 2 alleles for every gene. So if we have 2 alleles for each gene, what alleles we can have? Well, we can have big A, or big A, little a, or little a, little a. Now, homozygous dominant is a lethal allele. Heterozygous, it's a person would have achondroplasia, and homozygous recessive, that's a normal height, right? So now look, if we have 2 parents, both are with mutant gene, heterozygous, so they achondroplastic dwarf. Now, this is the Punnett square that tell us that we would have big A, 25%, big A, little a, 50%, and little a, 50%, right? So we should have like one, let's say if we have 4 offsprings, right? Like 4 kids, 1 should be normal height, 2 should have dwarfism, but 1 over here that is homozygous dominant, this is lethal allele, so this embryo will never be developed into a person. So the ratio is supposed to be 1 to 2 to 1, but actually of survivors, the ratio is different because this embryo doesn't survive. So the ratio is 2/3 to 1/3, and this is not a ratio that Mendel predicted us. So it seems like, oh, look, if we only study the survivors, right? We cannot study embryons in this example. Maybe those embryons were, they died even before mother knew that she was pregnant. So we cannot really study this embryo that might die like when they 5 days old only. So if you only look at survivors, we would say, whoa, look, this contradicts Mendel's law. But if we take into consideration those lethal alleles, we know it's follow exactly the same patterns. How do we know it if we cannot study embryons? Well, we can use this cross when we have a person with normal height and with achondroplasia. We make Punnett square, and then this works because we don't have lethal allele. We do have allele, but we don't have lethal genotype, homozygous dominant. So we would have 50-50, right? We will have 50% of kids who are a dwarf and 50% kids with normal height. So this cross shows us, yes, this follow this inheritance follow exactly the same pattern as Mendel predicted. Another example, multiple alleles. Individual carries 2 alleles for each autosomal gene. We already know it, right? If we are sexually reproducing organism, if you have 2 parents, then we'll get half of our genotype from one parent and half of genotype from another parent. However, genes can have multiple alleles, not just 2, because its sequence can deviate in many ways. Different allele combination can produce variations in a phenotype. An individual with 2 different mutant recessive alleles for the same gene is a compound heterozygote. So now let's look at the example to see how it works. So here's our multiple alleles example. In this example, we have 3 alleles. We have allele capital C, we have allele little c, and we have allele C, CH, and CH. And all these, so it's let me see how many we have. We have C, right? Capital C, CH. Oh, I'm sorry. Yes, CH, and we have C. So we have 4 different alleles for color in a rabbit, and different combination of this allele, because as we have rabbits over here, now this rabbit can only have 2 of these. This rabbit can only have 2 of these, but you have lots of variation. You choose between 4, right? If this rabbit is big C, then it's a black color. If rabbit is CH, then it's going to be a chinchilla color or Himalayan color or albino, right? So that's how multiple alleles affect phenotype. Another example of inheritance that seems that it doesn't follow Mendel's principle is incomplete dominance. When heterozygous phenotype is intermediate between those of 2 homozygous. So if you have a red flower and it's big R, homozygous dominant, and a white flower, homozygous recessive, and if red is dominant over white, then all big R, little r should be red. But in this example, all F1 generation gonna be pink. So why is it pink? Because red is not completely dominant, it's incompletely dominant or incomplete dominance patterns, exhibits incomplete dominance over white. So if it's not completely dominant, you will have intermediate phenotype. Another example of incomplete dominance is familial hypercholesterolemia. It's high cholesterol level in the blood. Now, heterozygous has approximately half the normal numbers of receptors in the liver for LDL cholesterol, and homozygous for the mutant allele totally lack of receptors, and so the serum cholesterol level is very high. Now, what it says that if you are homozygous for ability to make LDL receptors, so if you big H, then on your liver cells, so that's a cell of your liver, you have receptors. And what these receptors do, they attach this cholesterol to the receptors, and this cholesterol moves inside liver cells. So your blood cholesterol is low, right? So what does it mean people have high cholesterol level? Cholesterol level where? In their blood. So they draw their blood, they do the analysis of the blood, and they tell you how much cholesterol is in the serum of your blood. But these receptors allow to remove cholesterol from the blood inside the cells, and then metabolize it, get rid of those cholesterol. So if a person, a homozygous dominant, it has normal ability to make receptors. If it's homozygous recessive, look, no receptors. That means all this cholesterol stays in bloodstream. But heterozygous, because it's incomplete dominance, it should be, if it's complete dominance, it should be normal again. But no, in this example, the person only have half of a number of the receptors. So some cholesterol will be removed from the bloodstream, but not as much as we need to. So if we look at the distribution here, plasma cholesterol level in the general population, it should be below 300, but we have people who are between 300 and 600, right? Those are heterozygous for hypercholesterolemia. And if a person is homozygous for FH, then look how high the level of cholesterol in their blood is. So that's again incomplete dominance. Codominance, when we have heterozygous phenotypes that result from the expression of both alleles. Now, in this example, we have allele for blood type, allele A, allele B, and allele O. And I'm gonna just move to another slide to explain how it works. Now, if a person blood type A, I'm gonna draw it so I don't want here. So if you blood type A, you can be A. So we have three alleles. Let's start with alleles. You have allele A, allele B, and allele O. Now, you see, we have more than two, A, B, and O. But as a person, out of the three, you will only get two, because you will get one from mom, one from dad. So you cannot get all three at the same time, right? So if you blood A, you can be AA or AO, right? If you blood type B, you can be BB or BO, right? So let's say you get A from mom, A from dad, then you genotype AA. If you get A from mom and O from dad, you AO, you still type A, because O is recessive. But if you type O, you only can be OO. But look over here, if you A, you get A from mom and B from dad, neither one is dominant over another one, as a codominant. So your blood type is AB. This is the example of codominance. And this is the Punnett square that tells you that if a parent's, for example, if a mom is AA and dad is BB, and look at the children, and telling you 100% that kid's gonna be AB. So neither one of kids will inherit mom on that type, because alleles are codominant, so kids gonna be all AB. Right, so that's pretty interesting. Or if over here, for example, if mom type A and dad type B, but they AO and BO, the child can be O, the child can be AB, child can be B, child can be A. So that's a lot of variation because those are multiple alleles, and the allele A and B are codominant allele. Another process that can affect the pattern of inheritance is epistasis. It's a phenomenon when one gene affects the expression of a second gene. An example, Bombay phenotype. So look here for our blood type, again, example. If mother is type A and the father is type AB, now look, if mother is A, even if you say mother is AO, right, and father is AB, so look at our Punnett square, right? So this, let me clear it. So mother is AO and father is AB. The Punnett square tells us the child can be AA, AB, AO, and BO, right? Now, AO and BO is A. AO is A and BO is B. So child can be A, right? Child can be type B, and child can be type B, AB, because O is recessive. So here's what Mendel tells us. Well, Mendel didn't tell us that, but this is what we figure out based on his laws. So if a mother is type A, even if it's a heterozygous, and father is type AB, the child can be type A, type B, or type AB. And all of a sudden, we have child type O. How that happened? Well, that because we have another allele. It's called H allele. And if a child, another allele that affects the expression of allele A and B, and if a child has little h genotype, then this little a genotype phenotypes all gonna be type O. So you can be type A, B, or O, you're still gonna be only O, because what this gene does, it allows to this antigen A and B be attached to the blood cells. Let's look to the previous picture. To be type A, you need this glycoproteins A to be attached to the surface of your cells. You need glycoproteins type B to be attached to your cells. Now, this attachment, you need protein H to attach. So if you have little h, this cannot just attach. So you're gonna be like this. Doesn't matter what your genes are because of this epistasis. So many, of course, variation in our inheritance. And pleiotropy, it's a phenomenon when one gene controls several functions or has more than one effect. Can be difficult to trace through families because people with different subsets of symptoms may appear to have different diseases. Now, pleiotropy is shown over here. So, for example, that's our chromosome, and we have like a single gene that responsible for, in this example, beta globulin gene mutation. And because it's affect the structure of protein, one protein, it gives you different effects. It can affect your eyes, and the person is born, for example, blind because the mutation in this gene. But it's also important for liver function, so the person can be born with liver failure or with heart attack. So multiple traits, it's when one gene controls function of most, controls function of many organs or have more than one effect on the organs. All right, so another example of pleiotropy is Marfan syndrome, when a defect in the elastic connective tissue proteins called fibrillin cause symptoms in different organs. So here's a healthy person, and it's a chromosome 15, and we have a gene that code for protein fibrillin, and if it's not mutated gene, it will be transcribed and translated into working proteins. So you see that's a normal working proteins. However, if you have mutation in this gene, then this fibrillin is not assembled correctly, right? So we go from gene to protein, and then tissue that made of this protein is affected. But fibrillin found in many organs. They found in your lens, so it's going to be lens discoloration, can be long limbs, spindly fingers, caved in chest. So that's all Marfan syndrome. Because again, this is a protein that found in many connective tissue of your organ. And the most serious symptom is a weakening of aorta and sudden burst of the aorta that can cause death. And this is again why, because aorta also has this protein, fibrillin. So that's a pleiotropy. And again, pleiotropy, when one gene has effect on multiple organs. Genetic heterogeneity. Different genes can produce different phenotypes. Gene may encode enzymes that catalyze the same biochemical pathway or different proteins that are part of the pathway. So not only we have over here, we have a pleiotrophy. When one gene affects several organs, we also have the genetic heterogeneity when many genes produce identical phenotypes. So example would be osteogenesis imperfecta. That's a disease when a person doesn't produce normal protein collagen. Collagen is the protein that's very important protein in many organs. And if the production of collagen is altered because of different genes, because different genes produce all intermediate enzymes that catalyze the process of collagen formation, then we have at least 8 genetically distinct forms of the osteogenesis imperfecta. And retinal dystrophy is another example caused by mutation in more than 20 genes. So you have 20 genes, right, that work together to make sure that you have normal vision. If one of these genes has a mutation, so let's say I have mutation in gene 1, you have mutation in gene 2, somebody has mutation in gene 3, those people can be blind, but not because of the same mutation in the same gene, right? That's what our genetic heterotropy is. And that's what show us many routes of blindness. So you can either pigmental epithelium of the retina, it might affect rods, it might affect cones, right? So or it might affect some other proteins and other structures that passes the vision from your eyes to your brain. So, and when it's when you have alteration at any point that is responsible for your vision, then you know, you are blind or somebody is blind, but not so. What it's trying to say over here, blindness has many roots, right? It doesn't necessarily one gene, one mutation. It can be several genes and several different mutations, but it will be the same result. Person cannot see, right? So, and yes, so that's it for our first lecture, and we will pick up next time where we stop today. So next time we will talk about DNA of mitochondria. Okay. Thank you for watching. I want to remind you that this lecture trying to explain inheritance beyond Mendelian's law, and I hope it was helpful.